Hi, and welcome back to Microbiology. Today we're starting Chapter 11, Mechanisms of Microbial Genetics. Chapter 11 is a big one. It's going to cover everything from how DNA stores information to how it's transcribed, translated, and even regulated. In this first video, we're going to cover sections 11.1 and the first part of 11.2, the process of DNA replication. These are going to be the foundations for everything that comes later, like transcription, translation, and gene regulation. By the end of our first section, 11.1, you should be able to explain the two major functions of the genome, understand the central dogma of molecular biology, and distinguish between genotype and phenotype, and describe how the environment affects them. So Maimonides, who won the Nobel Prize in 1965 for his discoveries concerning genetic control of enzyme and virus synthesis, said this famous quote, "What is true in E. coli here is true in the elephant." And this really suggests that the biochemistry of life is shared in all forms of known life, right? All cells use DNA for information storage, we share the same genetic code, and we use similar mechanisms to replicate our DNA and express our genes. DNA has two essential roles. It stores and transmits hereditary information from one generation to the next. That means every new cell inherits the same genetic instructions. It also directs the synthesis of proteins that allow cells to grow, divide, and respond to their environment. And to do this accurately, DNA must be replicated with extremely high fidelity. Errors here could change how a cell functions. A gene is simply a segment of DNA that's transcribed into RNA. Messenger RNA, or mRNA, then carries the instructions for building a protein. Remember that transcription converts DNA to RNA, and then translation converts RNA to protein. The amino acid sequence of a protein determines how that protein folds and what it does in the cell. And together, these processes, transcription and translation, form or make up gene expression. The central dogma of molecular biology summarizes this flow of information. It tells us that DNA becomes RNA, becomes protein. So that's also seen here. Each step, such as transcription and translation, includes three substeps: initiation, elongation, and termination. This concept connects genotype, the genetic code, to phenotype, what we see as traits. And different patterns of gene expression produce the differences among cells, tissues, and organisms. The genotype is the full collection of genes in a cell. The phenotype is what we can observe, right? What we see. The traits or characteristics that result from protein activity. And even if two cells have the same genotype, their phenotypes can be different depending on which genes are turned on or off at that time. So gene regulation helps cells use energy efficiently and respond to their environment. And in the picture below, we can see that a single genotype can lead to two different phenotypes, these two different outcomes, depending on the environmental conditions at the time. So environmental conditions like temperature, nutrient availability, or stress can change which genes are expressed. For example, bacteria might activate stress response genes when they're exposed to antibiotics, and this flexibility allows cells to adapt and survive in changing conditions. One of the most famous relevant studies is an older one from 2019, the NASA twin study. And this revealed that astronaut Scott Kelly's gene expression changed while he was in space for about 340 days, compared to his twin brother Mark, who stayed on Earth. And it was really interesting because while most of these gene expression changes reverted to baseline, so they reverted to normal after he returned to Earth, there was still a small subset, like about 7% or so, that persisted even 6 months after he returned to Earth. That indicates there is some kind of definite body reaction to that spaceflight environment, and there could be a potential impact on human health for long-term space travel. As genome sequencing becomes faster and cheaper, we're really learning how genetic differences can affect susceptibility or our vulnerability to infection and treatment response. People are different. A pathogen that infects one person and causes major disease might not affect someone else. So someone else could be an asymptomatic carrier. So this raises ethical questions as well, because if we're studying genome sequences, we're studying someone's genetic sequence, we want to question if this genetic data should be shared, as well as who owns this data. So in 2008, Congress passed the GINA, Genetic Information Non-Discrimination Act, that protects people from discrimination by health insurers and employers, but it does not cover life or disability insurance. So these debates really highlight how science and ethics have to evolve together. There are statewide differences in these protections. For example, in 2022, California passed something known as California Genetic Information Privacy Act, or GIPA, G-I-P-A, and this imposed additional privacy and consent requirements on direct-to-consumer genetic testing companies. So direct-to-consumer testing companies include companies like 23andMe, Ancestry DNA, and many others. All right, in this checkpoint, they ask, what are two functions of DNA? At the beginning of the section, we learned about information storage and inheritance. So DNA carries the genetic instructions passed from parent to offspring. The second function would be control of cellular activity, right? DNA directs and regulates the production of proteins that are needed for cell growth, structure, and reproduction. For the second one, genotype versus phenotype. Genotype is the complete set of genes or genetic information in a cell, whereas the phenotype is the observable traits or characteristics that result from the proteins that are produced by those genes. And the third one, how can you have the same genotype but differ in their phenotype? Remember that the cells may express different set of genes depending on their environment or internal cell signals. Environmental factors such as temperature, nutrients, or stress can turn certain genes on or off. So as a result, genotypes can produce different phenotypes through changes in gene expression patterns. All right, and now let's also recap 11.1. So in the first one, two functions of the genome. The genome stores all genetic information in a cell. It contains every gene and regulatory region. So the two main functions are one, inheritance, transmitting that genetic information faithfully from parent to offspring through DNA replication. And two, gene expression, right, directing the synthesis of proteins that control cell structure, metabolism, and reproduction. For the second one, central dogma of molecular biology. So the central dogma describes the flow of genetic information within a cell, and it tells us that DNA becomes RNA, and then RNA becomes protein. So DNA is transcribed into RNA, RNA is translated into protein, and the sequence explains how genes determine the structure and function of proteins, linking genotype to phenotype. For the third one, similar to what I mentioned earlier, genotype is the complete set of genes or DNA that an organism or cell possesses, whereas the phenotype is what we see, right? The observable traits or characteristics produced by the proteins expressed from those genes. And there is an environmental influence, right? Different environmental conditions like temperature, nutrients, or stress can turn genes on or off through regulation of gene expression. So cells with the same genotype can exhibit different phenotypes depending on which genes are active during specific conditions. Now let's move into section 11.2 on DNA replication. This is the process that allows cells to copy their genetic material before cell division occurs. And replication must be accurate and fast to preserve the genome across generations. DNA replication is semi-conservative. That means each daughter molecule will keep one old strand and one new strand of DNA. So I see that over here in this image right here. So this is the original double-stranded DNA molecule, the parent DNA, and then here are my two daughter DNA molecules. For each one, I can see it's composed of an old strand as well as the new strand in red. DNA replication is also bidirectional. So we'll see later, I just basically turned this DNA strand sideways to see this image. DNA replication will cause an opening in the double DNA strand and will proceed bidirectionally in both directions. So this will involve a bunch of enzymes working together for both precision and speed. Historically, scientists proposed three models of DNA replication: conservative, semi-conservative, and dispersive. We now know, thanks to a famous experiment we'll talk about next, that the middle one is correct. Semi-conservative replication is correct. And that famous experiment was the Michelson and Stahl experiment. In 1958, these scientists grew E. coli in heavy nitrogen, nitrogen 15, and then switched to light nitrogen, nitrogen 14. And I'm going to use the next picture to describe the next steps. Remember that there were three hypotheses for DNA replication at the time: conservative, semi-conservative, or dispersive, and they wanted to figure out which one was correct. They used nitrogen since nitrogen is found in the nitrogenous bases of DNA. Remember A, G, T, and C. So E. coli was first grown in heavy nitrogen, nitrogen 15. It's heavy, so it sinks to the bottom of the tube after you spin it down through centrifugation. So this is the original DNA, and it's grown in heavy nitrogen, so it makes sense that they would see a heavy band in their tubes. Then they moved the E. coli to a lighter solution of nitrogen, nitrogen 14, and allowed the first replication cycle for DNA to occur. And nowadays we know that this would happen, but back then they weren't sure. So they decided, came up with what they should see for each of these types of replication. If replication was conservative, you would expect to see a heavy band, this blue one, and a light band, this red one. If replication was semi-conservative or dispersive, you would see intermediate bands between the heavy and light, because each new daughter strand of DNA, daughter molecule of DNA, is made up of both heavy and light. And that's actually what they saw. They saw a hybrid band and not this first scenario, that first possibility. So they eliminated the possibility of conservative replication. Then they allowed the E. coli to continue to grow in nitrogen 14, that light nitrogen. And they looked at what they would expect to see after that second round of replication. Again, we don't care about this one because we already eliminated that. If replication was semi-conservative, let me change colors because it's getting messy. If replication was semi-conservative, we would expect to see a hybrid band for those two DNA molecules and a light band for those daughter molecules. And that would look like this, the hybrid and the light band. But if DNA replication was dispersive, you would see only hybrid bands. And what they actually saw was that middle scenario, the hybrid and light band. They did not see this, only a hybrid band. So based on these results, they concluded that semi-conservative replication is what happens when you replicate DNA. This checkpoint asks, what would have been the conclusion of Michelson and Stahl if after the first generation they found two bands of DNA? So if two distinct bands, heavy and light, had appeared after one generation, after one round of replication, it would have supported the conservative model of replication, meaning the original parent DNA stayed completely intact and a brand new DNA was made from scratch. But what they actually found was that hybrid band, and then ultimately they found a hybrid and light band after the second round. So remember, their final conclusion was that DNA replication is semi-conservative. Before we dive into the steps for DNA replication, I wanted to remind everyone that nucleotides are the monomers of DNA, and we actually start with nucleotides that have 3 phosphates attached. So these are triphosphate deoxyribonucleotides. Deoxyribo refers to that deoxyribose sugar in the middle of the structure. And guanosine refers to the fact that this must be guanine. That's the nitrogenous base attached here. Remember also that DNA replication occurs with a lot of energy input, and that energy comes from these triphosphate deoxyribonucleotides themselves. Every time we attach our nucleotides to the growing chain of DNA, this bond is cleaved, and that breaking of this bond releases energy to power the building of our polynucleotide, our DNA molecule. Remember also that in the deoxyribose sugar, there are carbons, and the carbons are numbered. This is carbon number 1, 2, 3, 4, and 5 for our deoxyribose sugar. And you can only grow DNA by adding additional nucleotides on the 3' end. Let's start by looking at DNA replication in bacteria. So replication for E. coli, for instance, begins at an area called the ORIC, O-R-I-C. ORI stands for origin of replication. When we have the C here, ORIC, this stands for the origin of chromosomal replication, and that differentiates this type of replication from replicating plasmids or other types of DNA. And there are several enzymes that coordinate the start of DNA replication. We have DNA gyrase, or also known as topoisomerase 2, to relieve supercoiling. Remember, DNA is double-stranded and has a helix like this. And what you're going to have to do if I examine the DNA, I'm going to uncoil it. You're going to have to open this up, and that can accidentally over-tighten the ends. So gyrase will loosen up these ends to prevent the DNA from breaking. Helicase, so we have our double-stranded DNA, is going to break the hydrogen bonds connecting the bases. So at the end, you'll see a bubble form after helicase is finished breaking those hydrogen bonds. To prevent the DNA molecule from simply collapsing and reforming those hydrogen bonds, we have single-stranded binding proteins. And these look like little dots here that connect to that single-stranded DNA to prevent them from coming back together. So you're going to prevent that. Then we have an enzyme, RNA primase, that comes in. So let me redraw that. RNA primase is going to come in and put down a little molecule called RNA primer to allow DNA replication to begin. Remember, DNA replication has to start from the 3' end of the hydroxyl group of that deoxyribonucleotide. But if there's no 3' end, then the enzyme that does DNA replication, DNA polymerase 3, can't do anything. So RNA primase's job is to lay down a short molecule called the RNA primer that I'm showing in that dark red right here, in order for DNA polymerase 3 to have a 3' end to start replicating DNA. The next few slides will have screenshots from this YouTube video, which I'm going to post on Canvas because it has a great summary of DNA replication in prokaryotes. But I wanted to show you that this would be the origin of replication, and we have our replication bubble here. At the ends, that little kind of V shape or fork shape, those are called replication forks. Because they kind of look like the prongs of a fork. And to prevent our replication bubble from collapsing upon itself, we have our single-stranded binding proteins. The enzyme that separates the double-stranded DNA, so breaks those hydrogen bonds between the bases, is DNA helicase, and it's shown in green here. Since DNA replication cannot start from scratch, DNA polymerase 3, this is because DNA polymerase 3 requires a 3' end to exist before it can put down the next nucleotide. Since at first there's nothing there, primase, the enzyme RNA primase, has to put down an RNA primer in order to provide that 3' end for DNA polymerase number 3. So these red nucleotides are actually RNA nucleotides. So they have a ribose sugar instead of deoxyribose sugar. So if I have my DNA template here in blue and I arbitrarily put, let's say this is A, this is G, and this is C for my DNA nucleotides, my RNA primer in red would have to be complementary. So let me switch colors. What color can I use? Maybe I'll just stay red. This would be U, or actually I switched to blue, sorry. U, C, and G. That's my RNA primer. And then finally, because of that primer, I have a 3' hydroxyl group that my DNA polymerase number 3 can continue adding DNA nucleotides on. And the same thing is happening over here. So primase laid down a primer, DNA polymerase 3 continued adding DNA nucleotides after the primer, and it uses this DNA molecule as a template to do so. One thing to note is that since DNA replication can only go from 5 to 3, this must be the 5' end, and this end must be the 3' end. So it's growing in this direction for that new strand of DNA. The same thing must be happening here. That primer creates that 5' end, and it's growing along the 3' end. Because the template strand has to be opposite, remember DNA is anti-parallel, that means for this dark blue DNA molecule, this must be 3', and over here, this must be the 5' end. The same thing is true over here. If my new strand is 5 to 3, this dark blue original template strand must be 5' on this end and 3' on this end. Because DNA polymerase can only add nucleotides to the 3' end of a growing strand, this means that one strand, called the leading strand, will be built continuously toward the replication fork. There will be, and I'll show you soon, another strand called the lagging strand that is synthesized discontinuously away from the replication fork. These are going to be built in short pieces known as Okazaki fragments. There's also a protein called the sliding clamp that is important because it keeps DNA polymerase 3 attached to the DNA template for efficient synthesis of DNA. Otherwise, DNA polymerase 3 sometimes tends to fall off the template strand, and you need to take additional time for it to come back on. Let's look more closely at that orientation again. So I'm going to look at my original parent DNA double-stranded molecule first. The bottom, I said earlier, runs from 5 to 3, and then the other strand must be anti-parallel to that strand. Let me erase these random marks over here. So that means down here somewhere is the 3' end. Down here it's cut off, but that would be the 5' end. And again, I'm just orienting myself, looking at the original orientation of the parent strand of DNA, the template strand in dark blue. That tells me about the daughter strands. So that means my new strand of DNA that I'm making must be produced going this way, with that being the 5', this being the 3'. I should have given myself more space. That means the other side, that would be the 5', and this would be the 3'. All right, so the red is that new daughter strand. So this would be the 5' end, and DNA polymerase 3 is adding new DNA nucleotides to the 3' end. The same thing would be happening here, although it's cut off. It would be on the far left of the page, and I'm growing in this direction with the 3' end. My DNA polymerase 3 is adding new DNA nucleotides. While it's pushing forward, it also kicks off those single-stranded binding proteins. These are the leading strands, right? These two, this one as well as that one. They are built continuously towards those replication forks. But as DNA helicase continues to unwind the DNA double-stranded helix, there are new single-stranded DNA molecules here. And I know that I can't just attach DNA nucleotides going in this direction, right? You can't add to the 5' end, you can only add to the 3' end. So to fix this problem, what happens is primase comes into play again and sets down a primer so that DNA polymerase 3 can continue generating the rest of the fragment here. And that would continue to happen. Primer would be placed down, and then more DNA nucleotides placed down using DNA polymerase 3. So these would form those Okazaki fragments, and these make up the lagging strand. And we can see a little bit more of that lagging strand over here as DNA helicase moves down and unwinds the DNA, leaving these single-stranded regions open. The primase will come down every once in a while to lay down the primer. I can see several regions of primer, generating these Okazaki fragments. So each primer, along with its DNA nucleotides, that would be an Okazaki fragments. Here's my DNA polymerase. Looks like it set down some of these DNA nucleotides, then it's going to go continue the same thing over here to generate more DNA nucleotides from that 3' end. And then there is another enzyme called DNA polymerase number 1 who has to come in to remove the RNA primer, because you don't want ribose and RNA nucleotides in your DNA molecule, right? That should be double-stranded DNA at the end of DNA replication. So DNA polymerase number 1 removes primer and replaces that with DNA. All right, so I just mentioned it's DNA polymerase number 1 that removes the RNA primer and fills it up with DNA, fills up those gaps with DNA. Then DNA ligase comes in and it seals the nicks between those Okazaki fragments. Topoisomerase is also involved to prevent overwinding at the edges of the fork. So remember I showed you kind of like a bubble. Topoisomerases will work on the edges to make sure there's no overwinding. And in prokaryotes, they have a single circular chromosome, so replication occurs bidirectionally. And once they're done, sometimes they are still stuck together. They're kind of still connect or interlocked. So topoisomerase number 4 can actually cut and separate the 2 rings and then reseal that cut in order to generate those 2 separate chromosomes. Since DNA gyrase and topoisomerase number 4 are unique to bacteria, we have a group of drugs called quinolone antibiotics, that specifically target these enzymes without harming human cells. This is a nice figure that kind of summarizes everything. In light blue is my original parental DNA, my template strands. This one runs from 5 to 3 on the top, and the bottom one runs from 5 to 3 this way. They're anti-parallel. When they open up, because of helicase, looks like helicase is here, forming that replication bubble. I also see a replication fork, that little V shape on the side. I know that I can have a leading strand. So there was a series of steps that happened before this picture was shown. We have, for example, primase, put down a primer. We have DNA polymerase 3 continuing that growth of the strand. Then DNA polymerase 1 remove the primer and replace that with DNA. We also have our lagging strand shown here. In the lagging strand, you're continuously forming new fragments. So I have my primase come down and put down a primer. Looks like primer is probably the little green one. And then I have my DNA polymerase number 3 come in and continue growing that strand. And then remember DNA polymerase 1 has to come in to remove the primer, and DNA ligase has to come and seal those Okazaki fragments together. And this is a nice table from our book to tell you the functions of all of the enzymes we just discussed. When it says exonuclease, it means that this enzyme cuts from the ends of the DNA. So remember, primers are going to be on the end, 5' end of the growing DNA strand. So exo refers to the fact that the enzyme cuts from the ends and not within the middle. So it's cutting from the ends of the DNA molecule. All right, our next checkpoint asks, which enzyme breaks the hydrogen bonds holding the 2 strands of DNA together so that replication can occur? So remember, it forms that bubble. It's helicase. So helicase breaks the hydrogen bonds between the nitrogenous base pairs, separating the 2 DNA strands to form those replication forks at the ends. For the second one, is it lagging or leading strand that's synthesized in the direction toward the opening of the replication fork? So the leading strand is synthesized continuously to the opening of the replication fork, either this side or it's going this side. And the lagging strand is synthesized discontinuously away from the replication fork. So if this is my bubble, it would be like here, 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 moving towards the center of the bubble, the widest part of the bubble. And the next one, which enzyme is responsible for removing the RNA primers? We said it was DNA polymerase number 1. So DNA polymerase 1 removes the RNA primers and replaces them with DNA nucleotides in bacteria. And we'll finish 11.2 in the next video, looking at replication in eukaryotes.